Good morning, good afternoon, and good evening to everyone, and welcome to this Thermo Fisher Scientific webinar series supported by the AZO Network. My name is Darius Sulam, and I'm very pleased to be your host for today's event titled Enabling Artificial Intelligence for Future STEM Applications. We're very fortunate to have Eric Van Kaplan with us. Eric will showcase how artificial intelligence can improve workflows through automation in transmission electron microscopy. We're also very fortunate to have Eric Van Kaplan with us. Eric will showcase how artificial intelligence can improve workflows through automation in transmission electron microscopy. And now, the moment we've all been waiting for, may I introduce Eric Van Kaplan.
Thank you, Darius. Full disclosure, I'm an electron microscopist who's learning about AI and how AI can improve electron microscopy. And so far, it's been fascinating. As an introduction, I'd like to discuss why we as a company are interested in developing AI-assisted automation. After that, I'll briefly describe the enabler. That's AutoScript. And then I'll get to the interesting part, the real-world applications. So when I look at the current challenges in electron microscopy that could benefit from automation, I believe they fit in two buckets. The first bucket here on this slide is enabling data acquisition beyond the capabilities of a human operator. You know, microscopy used to be pretty straightforward, and then we started getting all these options that were added, and pretty soon operators were having too few arms and hands to operate all the systems. So I've mentioned a few here. Multimodal data acquisition, a lot of systems that need to be operated at the same time. Low dose acquisition, where basically you do blindfold adjustments before you take your shots, but also 24/7 batch acquisition. Nobody's going to do that. And then finally, in situ data acquisition, where a lot of parameters need to be managed. The second bucket is helping with making sense of the acquired data. And there's two aspects to that. First, by improving the data, those are the left hand items, like live imaging of atoms in less than perfect conditions, or working with extremely noisy data, making sure that the data is statistically relevant. Or on the right hand side, by helping extracting relevant information about the sample. I mentioned there data mining and 4D STEM. When you do 4D STEM experiments, you're acquiring gigabytes of data. If you do a few of those in a day, you might hit a terabyte. And we need help on how to extract the valuable, relevant information. I also mentioned on-the-fly characterization, because if you have on-the-fly information, that'll impact how you do your experiment. And if conferences are a measure of what is coming, then I think that, judging from the latest conferences, automation is clearly the next EM frontier. I looked at M&M specifically, and I found over 30 talks on AI and automation, and I'm listing the topics up there. But in general, what I found was that the recurring challenges were training the neural networks and collecting the data. Now, I believe that we made significant progress in both of these areas, and that's what I'll try to demonstrate with the real-world examples that I'll be showing later on. But first, I need to talk about AutoScript, because it's AutoScript that is going to enable AI. We've had AutoScript for our SEM and dual beam products for many, many years, and now it has been extended to our TEM product line, meaning the Talos and the Spectra. AutoScript is a so-called API, an application programming interface, and the environment that we chose for API is Python. Why Python? Well, it's the fastest growing open source language. It's a great community. There's lots of tutorial and reference materials online. It easily interfaces with other languages. And for free, you get a large number of libraries. And on the lower right-hand side of the slide, you see just a quick example on how to acquire and save a stem image. AutoScript has two components. It's got the server part and the client part. The server part is hosted by the microscope itself. It's the microscope PC. And the left-hand side of the slide shows the Thermo Fisher components. And the right-hand side shows the Python ecosystem. There are two scripting scenarios. So the AutoScript server is hosted on the microscope PC itself. And in the default setup, the AutoScript client is a support PC or, your laptop that interfaces with that. And that's how you do the scripting. There's an alternative solution, the local solution that probably arises during testing. And that's where you do everything on the microscope PC. So those are the two scenarios to do scripting. Okay, let's change gears and go to the real world AI assisted automation examples that I have here today. The first one is actually about the Thermo Fisher Scientific Metrios. It's a dedicated TEM stem, fully automated for the semiconductor industry. Now, this is not a presentation about the Metrios, so if you want to know more about it, I encourage you to look at a website. Here, what I'm doing is highlighting the artificial intelligence components in the Metrios. In general, the biggest automation challenge is to recognize features. It's based on fully convolutional neural networks, or FCNs, and that is, in this case, Metrios based. The first thing you have to do is basically train the neural networks. And let's look at an actual exercise here, finding gate-all-around or GAA logic transistors in an SRAM cell. The first thing is you need to tell the system what a fib lamella is. In this case, it's a top-down wafer preparation. And so the system needs to know what, at low magnification, what a lamella looks like in order to be able to zoom in and then discover the device line. Once you have the device line, you further zoom in, you tilt the stage, you move the stage, and you then tell the system what the GAAs are. The GAAs are these squarish, roundish, white circles that you see there. And the white stuff is the gate itself, and it's all around. Once the system knows how to do this, it'll do its magic, and then in subsequent samples will tell you, well, hey, I discovered a lamella. It zooms in and says, haha, I did find the device line. And then finally, it'll measure the devices that it was thought to measure, and the returns will be a bunch of coordinates. Obviously, this is only the starting point, but again, this is not a presentation about Metrios. This is the starting point to do, let's say, critical dimension measurements, etc. Right. So, big names, artificial intelligence, deep learning, machine learning, neural networks. Well, what does that actually mean in the field of, in the framework of electron microscopy? Well, most commonly, the input will be a grayscale image, like on the left-hand side, where we see a 3D NAND, that's memory, and each little circle there is a bit in your memory cell. That's the input. The output is a bunch of coordinates because you want to know where those bits are. So the output is a segmented map. And how do you get from left to right? Well, that's where we use the neural networks. And that is the stuff that's inspired by your visual cortex in the brain. So once you've trained the network to look for something, it'll start applying filters. It has a bag of tricks, and it will exhaust that bag of tricks until it detects the trained objects 100%. So in general, this is done by kernels. So it's sweeping kernels over the image. So it starts with a 2x2 kernel, then a 3x3, 4x4, and so on. It uses, in combination, all the filters it knows about. And then it, on its own, decides what the best method is to basically detect 100% of the objects that you told it to discover. And that's basically why it's called artificial intelligence or machine learning, because at the end of the day, no one really knows how the neural network arrived to the output. We've given it some functionality, but we don't know exactly, in this case, in this particular case, we don't know exactly what it did. And that's why it's called hidden layers. And there are, most of the time, multiple hidden layers, and that's where the word deep learning then comes from. Okay. Here, just an example of what it can do. So in short, neural networks segment grayscale images so as to extract features of interest. And the output, this output can then be used to just do image acquisition. But the same output can then also be the starting point for performing measurements on the object. Again, this is outside the topic of the presentation today. How does AI compare to conventional pattern recognition algorithms? So here we use a bright field TEM image of GAAs, gate-all-arounds. And on the left-hand side, we used a software package called Cognex, and we had to put in a bunch of parameters to try to explain what gate-all-arounds are. And it found, in this case, about 22% of the gate-all-arounds. Why? Because the other ones are distorted, bigger, smaller, whatever. And obviously, by playing with the parameters, you can crank up that number. Maybe you can get to 30%, 40%. But that's going to be very time consuming. On the right-hand side, a neural network, and it found 100% of the GAAs, whether they were distorted, complete, bigger or smaller. It did 100% discovery. So what does segmentation look like? How do you basically train the network? And here's an example of an operator teaching a neural network what a gate fin structure in a transistor looks like. And he does it both on bright field images as here, and later on he'll do it on a dark field image. And also notice that he's not doing a perfect job. He is approximately delineating the fin gate structures. And here he's doing it on a dark field image. But just like your brain, as the neural network will finally grasp what to look for as long as you give it sufficient examples. So that's why it doesn't have to be perfect, but give it sufficient examples and it will finally totally understand what it's looking for. And then notice on the left-hand side, and probably there at the right-hand side also, where there's a partial fin, he also explains the system to look for partial fins, so they will also be counted. So, but that's basically it, and he looks at all the examples he's given it, and that should be sufficient for the neural network to now discover on its own. Let's move on now. Let's go from semiconductor to material science. So basically from a commercial Thermo Fisher product to customized automation developed by early adopters of the AI functionality on our TEMs. Both examples are about so-called adaptive scanning. The first one is explaining what adaptive scanning is, and this approach was developed by BASF in Germany, and they kindly allow me to use their results. They have a Talos TM for catalysis, and obviously time to data and statistically meaningful data are paramount for them. So what they do is they first take a fast overview like here. So they quick, fast HADF image, and it's nanoparticles on a porous substrate. And then they use a neural network that was properly trained to detect all the particles on the substrate. And so they run the neural networks, and then they get the green dots here, which are all the particles from the left-hand side image. Then EDX spectra are acquired just on the green pixels. So now you go back into your STEM image, the green pixels, you do acquire data, and from those spectra, you can now colorize the atoms according to their composition. Obviously, this will save a vast amount of time compared to doing an EDX map of the whole region, but it will also improve the integrity of the sample because porous substrates are known to degrade under irradiation. So now the fact that you used way less electrons to acquire the same data, the integrity of the sample is preserved. Let's look into a little more detail how this works. So again, porous material with nanoparticles, a quick overview image. Launch the neural networks to discover where the nanoparticles are. From there, we can do some shape interpolation, so we get information about the shape of the nanoparticles, and we can also generate a scanning mask. Now, from the shape interpolation, we can extract shape statistics. From the scanning masks, we can acquire EDX spectra. From the EDX spectra, we get the composition. And finally, shape statistics and composition can then be combined into the final report. So what does BASF gain from this? Well, here is an example. 3x3 HADF stitched tiles. And in those 9 HADF stitched tiles, they've discovered 4,861 particles. They did shape analysis of these particles. They did compositional analysis of these particles. And the whole thing, acquisition and analysis, took about 23 minutes. Obviously, a conventional approach to analyze 4,861 particles would have been prohibitively long. As a matter of fact, just the EDX acquisition would have taken 25 hours. Let's move on to another example that I call compositional information in HR STEM images. The question we're trying to answer here is, can we get compositional information with just a fraction of the dose of a conventional EDX map? Obviously, the answer is yes, or otherwise I wouldn't be sitting here. But it's yes because we're using the fact that the world is discrete, that materials consist of atoms. And for testing purposes, the starting image, as you can see here, isn't a great HR STEM image. It's a low contrast strontium titanate image. But then we let loose a neural network which knows how to detect atom positions. And from these atom positions, we can then generate a dual speed pattern. What do I mean with dual speed? Well, in between the atoms, we scan with 3 microseconds background, and on the atoms with 3 milliseconds, so 1,000 times longer on the atoms than in between the atoms. From that, we can now do HADF and EDX, so HADF in between the atoms and EDX on the atoms. Obviously, from multiple scans, I can also derive the drift. From the drift, I can do drift-corrected EDX. So now, on the atom positions, I can acquire EDX for 3 milliseconds drift-corrected. From those spectra, I can now do atom identification. Obviously, initially, there's going to be a lot of mistakes because the statistics aren't good enough. So I'm looking at the error. If the error is too big, I continue. So I iterate this procedure until the error is below a certain threshold. So let's look into a little more detail. Here I have my poor strontium titanate HADF image. But notwithstanding the fact that it's a poor image, your brain manages to figure out where the atom positions are. We see those blobs. It's not a great image, but each and every of us can really tell where the atom positions are. Well, a well-trained neural network can do the exact same thing. So we launch a neural network, and this is the output, and we clearly see where the atom positions are. The little veil that you see on this image corresponds to lower statistics, and we can see that in the original image. So the unveiled areas clearly have a better contrast than the veiled areas, and that's just a way of telling the operator, hey, the precision is less in the veiled areas than in the unveiled areas. Now, from that atom probability prediction map, we can, through thresholding, come to a binary mask. And now we can tell that atoms are round, and we get this final map. So all the green dots are the coordinates of atom positions, and the red big circles represent big atoms, and the small blue circles represent small atoms. Now, that's what you see on the left-hand side. So the yellow dots are the atom centers, the red circles are the large atoms, and the blue circles are the small atoms. This image is acquired, it's a 512 by 512 image at 3 microseconds, and there's about 213 atom positions in this image. On the right-hand side, we see the exact same image. The second one here is an EDX acquisition. So in between the atoms, fast HADF, on the atoms, EDX. So now the whole frame only takes about 1.36 seconds. And with the next scan, I can then measure drift and so on. So all of these frames are 1.36 seconds in length. And through cross correlation, I can then figure out what the drift was. So now I'm going to false colorize the image. And here is the way I did it. When you see an empty circle, the identification was correct. So the big circles should be strontium, the small circles should be titanium. When EDX confirmed that, we keep the circles open. If there was a mismatch between the atom size and EDX, we fill it. So you could see blue circles filled with red dots. Those were misidentified. You can also see red circles with blue dots. They were also misidentified. And on the right-hand side, you see the full 213 atoms. So the first line, the second line, etc. So this is at the starting point after one frame of 1.3 seconds. Now, after 5 frames, the situation got significantly better. Now I only have like about 41 atoms that are misidentified. If I go to 10 frames, 17 atom columns are misidentified. 15, it drops. 20, I only have 2 columns misidentified. And at 22, everything's fine. At 30, we have 1 coming in again. And so above 30, nothing has been misidentified anymore. And so on the right upper corner, you see the error as function of number of frames. And what we see here is that basically after 22 frames of 1.3 seconds, which is under half a minute, we can actually stop the experiment because we will have correctly identified all the atoms. That means way, way less dose than for a conventional EDX pattern. So if I do a rough calculation here, I come to 550 times less dose than a conventional EDX pattern. I will not keep that number in mind. I'll just say that compositional info with drastically less dose. Obviously, this example isn't yielding new science. Strontium titanate is very well known and it can withstand very high electron doses. So we would have been able to do conventional EDX on there. But what this example does demonstrate is the potential of AI-assisted automation to drastically reduce total dose. And for some materials, this will be the difference between being able to get compositional information or not. So I'm now a big believer. AI-assisted automation will indeed transform electron microscopy. And not just for what the obvious reasons, not just for ease of use and time to data. It will also enable new science. And that then brings me to my last slide, the one introduced by Richard. The last slide is a conclusion slide with the 3 pillars that we're hoping to improve. Productivity. Besides the obvious boost from automation, I believe that AI promises to be much faster and much more robust than conventional approaches like pattern recognition. For the accessibility pillar, scientists and engineers can now spend more time doing their work, their research, and less time driving the TEM. In terms of confidence, we get the big picture. We get the big picture and we eliminate operator bias. And obviously, the 4th pillar that's not really mentioned here is the new science one. This is the end of my webinar. I'd like to thank everybody on the call for attending. And I believe that we're having a Q&A session now.
Fantastic. Thank you very much, Eric, for that excellent presentation. So the first question I have for you is, can AutoScript record the separate signals from the different segments of the STEM detectors separately?
Yes, it does. It does, and this allows, for instance, to do different differential phase contrast imaging or even integrated differential phase contrast. So yes, we have individual access to the different segments of the detector.
Great, thank you very much. Is it possible to automate the microscope with AutoScript?
Yes, as a matter of fact, AutoScript is the API that allows you to automate, and in that sense, it's only the enabler of AI. So that's the first step. You need full automated access to the microscope before you can launch other applications using AI. Yes.
Great. In slide 23, you show a very nice example of improving spatial resolution via AI on a SDEM image. But you also mentioned that it can be used to denoise images. Is it something that can be used as a low-dose image acquisition approach?
Yes, totally. And again, it's all about training. You need to train the system to look for atoms or preferably atom columns. That's what I should say. And once the system is trained, just like your brain, it'll see where the columns are. The thing is that once a machine is trained to do something, it ends up doing a better job than yourself, doesn't get tired, and it will find all the atom column positions. And once you have access to the atom column positions, it's then just a matter of visualization, in a more pleasing, denoised way. Yes. So yes, that's really, I believe, what I meant with new science. We will be able to actually use very, very noisy images and still extract valuable data.
Amazing, thank you. We have time just for one last question here. And so, what is the difference between the two scripting scenarios, local and remote? Which one of these is preferred?
Well, there's no real difference from a user perspective. You do the same thing whether you work from a remote position, a remote computer, and that can be a support PC, it can be a laptop that is connected to a support PC, or you work straight at the microscope itself. And so at the microscope, I can imagine that you'll do that when you're testing new functionality because you want to be close to the microscope to see that what you're programming is actually also happening. So I think that when you're in the testing phase, you'll be local. And once everything is tested, functionality is working, you'll be working remotely.
Amazing. Thank you so much, Eric, for all of your fantastic insights today, both for your presentation as well as the great Q&A session. Thank you.
Very welcome. Thank you very much, everybody.